All right, and I think we're live. So hello and welcome everyone to today's webinar titled Single Cells, Infinite Insights, Overcoming Obstacles for Top-Notch Data. So I'd like to welcome you all and also begin by thanking our speakers for today's webinar, as well as our sponsors, Singleron and Macrogen. Our speakers have a really fascinating story to tell, and I'm sure you'll find it both interesting and useful. But before I pass over to our speakers, a few quick things from me. So the first thing is questions. Feel free to ask questions whenever you like. You can use the questions tab on the lower right-hand side of your panel there. On demand. So if you miss something one of our speakers said, or you have to leave early, the webinar is available on demand. So you can watch the webinar back whenever you like. Just use the link in your registration email or go to our website at frontlinegenomics.com. The on-demand version becomes available just a few seconds after this live broadcast ends. So you can also stick around to watch it immediately. And if you would like one of your colleagues to watch, if you think it's worth them spending their time watching it, having you watched it, then they can also register to see the on-demand version after the webinar has finished as well. A quick note on polls. So we have a few polls, which I think the answers should be interesting for you. So we're going to ask you to answer a few questions as we go through, just 3 in total. Each poll only takes a moment, and you can access the polling questions by going to the polls tab on the bottom right-hand side of your screen. You can see it at the bottom there, just near the chat section. We will let you know when to answer each polling question. You won't see them until we make them available. But when we make them available, you can answer them, and it's nice and easy for you to do that. So on to today's webinar. Well, we've got 3 fantastic speakers who are all going to be part of telling a really fascinating story. So we have Luigina, Andrea, and Khalil. So I'm now going to hand over to Luigina, who will introduce herself. Over to you, Luigina.
Hi, hello, everybody. Thank you, Richard. It is a pleasure for me to be part of this webinar today, and I will tell this fantastic, fascinating story. And I will tell our experience in single-cell RNA sequencing analysis, and I hope this could be interesting and helpful for you. First of all, I can introduce myself. I am a biologist and a research scientist. I work at the pediatric hospital Burlo Garofalo in Trieste, in the northeast of Italy. And specifically, I work in the pediatric gastrointestinal unit of this hospital. Our main research topic is celiac disease. Celiac disease is an autoimmune disease induced by gluten ingestion, and gluten is contained in wheat, barley, and rye. And a patient with celiac disease suffers from intestinal and extraintestinal symptoms. In these patients, the ingestion of gluten induce an enteropathy characterized by the production of antibodies, in particular anti-tissue transglutaminase antibodies. These are autoantibodies produced at the intestinal level. In these patients, there is a problem of malabsorption because their intestinal tissue show an intestinal atrophy, so their gut doesn't work very good. This condition is related, as I already said, is related to the ingestion of gluten, and this condition can be diagnosed after gluten ingestion and can be diagnosed in children as well in adults when the symptomatology is not completely evident. The only treatment available is a strict and lifelong gluten-free diet. We know a lot of things about this disease, this condition. However, the pathogenesis is not yet fully understood, and there are still many unknowns. So, single-cell RNA-seq analysis could improve our understanding of this disease. And this analysis is a powerful tool for profiling and analyzing individual cell data, making it possible to resolve rare and intermediate cell states that are hardly observed at the bulk level. A study describing single-cell RNA sequencing profiles of intestinal tissue from patients with celiac disease is missing. We thought that this approach in celiac disease could offer a different view of this condition and could let us to identify new cell type typical of CD, or new target therapy, or new biomarker specific for celiac disease, or pathogenic pathways. Therefore, we decided to perform this study of single-cell RNA sequencing analysis on intestinal biopsies from patients. In the project design, we planned to enroll 10 pediatric subjects, and in particular 5 patients with celiac disease and 5 controls suffering from minor gastrointestinal disorders. From each enrolled patient, we will collect biopsies, and we will perform the single-cell RNA sequencing analysis. The workflow to perform single-cell RNA sequencing analysis include many steps: tissue sampling, tissue dissociation, library preparation and sequencing, and finally the bioinformatic analysis. Step 1 is tissue sampling, and it is very easy for us because I work in the gastrointestinal unit, as already said, and after collecting the informed consent, we can enroll the patient and collect intestinal biopsies. Then we have step 2, the tissue dissociation, and we have a well-established protocol to dissociate intestinal tissue. This protocol includes an enzymatic and mechanical dissociation by using liberase and hyaluronidase enzymes, by using a shaker at 37 degrees, and by pipetting. When the tissue is dissociated, we proceed with a filter passage to remove tissue residual and then centrifuge. The pellet is resuspended and filtered again to obtain a single-cell suspension. And then we have step 3, that is the library preparation and sequencing to obtain data useful for the bioinformatic analysis. In this workflow, step 3 is our limiting step. And probably we are not only facing challenging in this single-cell workflow. And this is the reason why there is a poll question for the audience to see what the most challenging step is for most users. In our case, we were not able to prepare and sequence the libraries in-house, and we need to outsource this step. Therefore, I contacted Macrogen Europe through Andrea Villino, and Andrea today is here with us, and I invite him to tell you this part of the story.
So thank you very much, Luigina, for the introduction. My name is Andrea Villino, and I am the NGS Senior Account Manager for Italy and Switzerland at Macrogen Europe. Indeed, Luigina contacted Macrogen Europe because We provide all types of sequencing services for research institutions like the Institute for Maternal and Child Health Burlo Garofalo. When Luigina contacted me about her single-cell project, we immediately scheduled an online consultation to understand the scope of her research. So basically, during the consultation and after understanding her project, Luigina expressed the interest in using the platform of 10x Chromium, which is commonly used for such cases. To proceed with this approach, we informed Luigina that she needed to send the samples in a frozen suspension cell using dry ice. Once of the risk involved was the freezing. The cells could compromise their viability, as good cells are quite fragile, as Luigina explained. This concern was confirmed, unfortunately, by the sample QC, which showed a cell viability of 40-50% and a low cell clustering. Therefore, considering this result of the sample QC, we decided not to proceed with this workflow. I really want to emphasize that this decision was made because the sample QC did not meet the necessary standards for further steps. So given the tissue origin of this and the sensitivity of the samples, freezing them for shipment proved to be too detrimental to their stability. And considering this result and the range of solution that Macrogen Europe can provide, we propose to Luigina a different platform and approach, Singleron. Considering her needs to ship samples, Singleron offers the option of sending fresh samples in a tissue preservation solution buffer that can preserve the tissue for up to 72 hours without requiring dry ice. So Luigina opted for this shipment workflow, and in less of 24 hours, the samples arrive at the Singleron facility. For the sample QC, Singleron provided the tissue dissociation and the sample QC, and according to the sample QC report, as you can see here, the cell viability was above 90%. After sharing this result and receiving approval from Luigina, Singleron proceeded with the constructing the libraries with their technology JEXscope and sent them to Macrogen Europe for sequencing. On the Novasec 6000 provided by Lumina, obtaining 90 gigabases per sample at throughput. So Kahil will explain in the next details of their technology. However, with this graph, I wanted to show the difference between the 10x and the Singleron. So basically, 10x use a droplet-mated method, while Singleron employs microwell-based method. However, the shipment was the key factor in this project. Due to the need to outsource this part of Luigina research and the nature of her samples, freezing them for shipment was not feasible. So therefore, utilizing Singleron tissue preservation buffer option for shipment was the best approach for this specific project. Now I am glad to give the floor to Kahil for the next slides.
Yeah, thank you, Andrea. And yeah, hi, my name is Khalil, and I'm heading the application support department here at Singleron. And as Andrea said, we offer solutions to make it easier for people to send us their samples, where we have gathered a lot of expertise, taken the sample from tissue all the way to data. And the part, the beginning, the first part, which is getting the single cell suspension, is for me as a person coming from bulk RNA, kind of something different. It's really an art in itself, taking a piece of tissue, dissociating it into a suspension of cells that is still viable so that you can get high quality data for it, and that it remains viable until you can isolate the cells individually, barcode them, and extract the RNA from them. And in this case, we have here as a company gathered a lot of experience across many species and many tissue types. And we offer even two ways of doing things. So we have the automated and we have a manual method. The automated, we do it with two devices. Here you can see a representation of them, which we call the Python. And the idea of tissue dissociation is really trying to get the individual cells separate by breaking down the cell-cell connection, but also the cell matrix connections, while preserving cellular representation. It makes no sense if I take a piece of tissue and get only the epithelial cells. So we need to get every type of cell from this tissue, and in a way that preserves the representation. So we need to have the relationship between the cells in the tissue in our single-cell suspension. At the same time, also, it needs to be harsh enough to dissociate all these cell types, but mild enough to keep them intact and not having them having a membrane leak and losing the genetic material or having the cells die. And speaking of that, also preventing a stress response. And yeah, so I don't know, we have a poll here, so maybe you can take some time and answer how is the tissue dissociation step for you? So I will maybe give you time to answer that poll. Okay, yeah. So I will continue speaking. And yeah, so the dissociation is basically, there are two types. So there's the enzymatic, and this is where our tissue dissociation buffers come in, as well as mechanical. And this is just by force, so pipetting or shaking on a shaker. What is offered also is in our Python, we have these tissue dissociation tubes where we put a piece of tissue with the dissociation buffer, and then in the machine where rotation of these plastic blades breaks down the tissue mechanically. We have a universal dissociation buffer, and this works with most tissues, but also, as I said, tissues are very, very different. And therefore, in some cases, we need a more specialized approach. And this is where we have several buffers for skin, pancreas, GI tract, where you have enzymes that might break down the cells and reduce the quality of the suspensions. And this is indeed, in Luigina's case, very important. And then we have a manual and automated, as I said, but our experience, and this helped us, like in this project, but also in other projects, where we have worked with more than 40 species. So I was looking at the table the other day, and it's amazing how much experience there is there. 400 tissues across those species, and also the multiple disease states. And each one of them changes stuff around. For example, as Luigina said, it's celiac disease, and this might be different from a normal intestine. And also the age plays a role. So whether you're working with mice that are young or old, that changes some things. For example, cartilage and bone, those are very hard tissues to work with. And the buffer that Andrea mentioned and Luigina also are important for customers to be able to transfer their tissue to us without having to freeze them. And therefore, we can do single cell instead of single nucleus. And yeah, with that, I would like to give the floor back to Luigina.
Thank you. So, as Khalil explained, we send the biopsies, and they were able to dissociate the samples and to generate the single cell RNA, the library, the single cell library. And then Macrogen performed the sequencing. So Macrogen sent us the data, and we performed the bioinformatic analysis. In both patients, the CD1 and the control one, we obtained transcriptional data from more than 5,000 single cells per sample. However, after quality filtering, a high percentage of cells was lost. In particular, this was related to the mitochondrial content that was very high in the majority of the analyzed cells. Usually, an high mitochondrial content is related to suffering cells. So at this point, we tried to find a way to improve and overcome this obstacle. We guessed that the transport of the tissue in the tissue preservation solution could be the limiting point and affect the quality of the data. And to explore and verify this hypothesis, we decided to take advantage from the fact that the single-run kit to prepare single-cell library can be performed by using the instrument, but it can be performed also manually. And we decided to use this kit in-house and to perform the protocol manually. So we collected the intestinal samples and from 2 more patients, and we dissociated the fresh tissue immediately after the collection, and then we prepared the library manually in-house. So we enrolled 2 more patients, again 1 patient suffering from celiac disease, and the other one was a control. We prepared the libraries manually. Macrogen performed the sequencing, and at the end we obtained the data from around 10,000 single cells per sample per patient. After quality filter, we lost an acceptable percentage of cells, and in particular, this time the mitochondrial content was acceptable. So probably the shipment of the intestinal tissue in the buffer was not a good option for this kind of tissue, or at least for our samples. So, since the data were of good quality, we started our bioinformatic analysis, and in particular, we proceeded with cell clustering analysis. And with this analysis, we found 22 clusters in both in the control patient and the patient with celiac disease. In each cluster, cells share the expression gene pattern. So in each cluster, there are similar kind of cells. And the 22 clusters were found in both in the celiac patient and in the control one. We then proceeded trying to identify these clusters, and we were able to identify all the 22 clusters, and so all cell clusters were named by identifying the genes specifically expressed in each cell type. Then, even if we have enrolled and have data from only 2 subjects, 1 patient with CD and 1 control, we performed a preliminary differential analysis. And thanks to this analysis, we have observed that cell clusters were represented in celiac patients and control patients at different frequencies. In particular, we observed an increased number of cells of the immune system. And these cells, this cluster are in orange, I think orange, yes. And we observed an increased number of these cells that are part of the immune system. Indeed, you can see that NK cells or CD4, macrophages, plasma cells, in particular IgA plasma cells, are increased in celiac patients in comparison to the control one. And this is in line with the disease, because this is an autoimmune disease in which the immune system is activated. In particular, the IgA plasma cells are activated because these are the cells able to produce the anti-tissue transglutaminase antibodies that represent the specific and typical marker of this disease. Moreover, we observed a reduced number of enterocytes that you can see in green, and enterocytes are the absorptive cells. So with this preliminary data, observed a reduced number of these cells. And also this result is in line with the condition, because as I already said, this is an intestinal condition in which the gut is not healthy, because there is an intestinal atrophy, and there is also and these patients have problem of malabsorption. So this confirms that the intestinal tissue is not healthy in these patients. And then we observed one new interesting thing that is about the goblet cells, because we have observed an increased number of goblet cells in the CD patients, suggesting an unexpected involvement of these cells in the pathogenesis of celiac disease. These no data are present in literature about these cells and the involvement of these cells in the pathogenesis of celiac disease. So this result, if we will confirm this result, we have to further investigate this aspect to understand why goblet cells are increased in this disease. So what we have learned from this story, that we have learned that the intestinal tissue is a tough tissue, and we cannot perform single-cell RNA sequencing analysis by dissociating this tissue in a cellular suspension and freezing this, because this tissue as cellular suspension is not viable after freezing. Moreover, we have learned that the intestinal tissue cannot be, in particular the samples and the intestinal tissue from celiac patient, is not preservable in tissue preservation solution. That is an option offered from Singleron. And so finally, we have to process to obtain good and data of quality. We have to process this tissue immediately to obtain top-notch single-cell RNA sequencing data. it is possible to perform single cell RNA sequencing in the intestinal tissue and obtain high quality single cell data. And this represents our first goal and aim achieved. Moreover, our preliminary analysis revealed that in the intestinal tissue there are at least 22 distinct cell types, because we found 22 cell clusters. And in particular, we have found an increased numbers of immune cells in the sample from the patient with celiac disease. And we also observed a loss of absorptive cells in the patients suffering from celiac disease. What about next steps? Now we are happy because we know how to realize this project, because we know that to generate single cell RNA sequencing libraries from intestinal tissue is feasible, and we can do it by ourselves in-house. And we will continue with this project. So we will complete the patient's enrollment and to obtain single cell RNA sequencing data from 5 patients with CB and 5 control, as described in the project design. And we will perform in-depth gene expression analysis to achieve the final goals, that are to identify specific celiac disease associated changes in intestinal tissue composition, to complete our understanding of the pathogenesis of this condition, to identify new therapeutic targets and biomarkers specific for this disease, to offer to these patients an alternative to the gluten-free diet that they have to conduct for all their life. And now I just want to thank you to all the people that are working to this ambitious project. And in particular, I say thank you to Fabiana Ziberna, that is a biologist working at the bench and preparing the libraries. And then I want to thank you, Michelangelo and Nicolò, because they are expert in bioinformatic analysis, and their work is fundamental to perform this kind of study. And then thank you also to Giuseppe Molinario, our PhD student that is working on this project, that is the study of his PhD. And thank you to all of you.
All right. That's great. Thank you very much, Luigina, Khalil, and Andrea. What a fascinating presentation. And I can see there are already quite a lot of questions. What we also have, however, is another poll. So I'm just going to take that poll live right now. Okay, so this poll question, before we get onto the Q&A, is just to help us understand what other topic would be of interest to you in a future webinar. Now, there are some options on there, but it's quite likely that you have answers that aren't covered in the options. So if there is an other topic that you would like to be considered, then just click the other box and then go into the chat section and just type the topic in. Or you could just type the topic in the chat section if you wanted to save a step there as well. So I'll just give you a few seconds to take a look and answer. All right, great. Thank you very much for that. Okay, so we've got quite a few questions. I'm going to start with the most upvoted question at the moment, which is from Flourish Adebayo, which is, what bioinformatics tool was used for downstream analysis? Who wants to take that one?
I don't know if, Luigina, did you do your analysis? I can't remember in this case.
Yeah, the analyses were performed by Michelangelo and Nicolò, so I'm not the right person to answer to this question, but I will ask them and we will answer to this question as soon as possible.
No problem. So what we can do, Flourish, is come back to you with an answer to that question directly. Brilliant. All right. Thank you, Luigina. So the next question is from Karim Fakir. Is the number of cells in the different condition have been normalized to be compared, as it can be difficult to compare if the starting number of cells is different? I don't know. I didn't quite get that question. So is the number of cells in the different conditions, have they been normalized to be compared? I think that's the question.
Yeah, I understand the question. It is not possible to normalize the number of cells. We normalize the cell by checking the quality. Unfortunately, as I said, this is a preliminary study, so we have data only from two patients, two different patients, one patient with celiac disease and one control. So we will continue our project to achieve our goal, that is to have 5 patients with celiac disease and 5 controls, and we hope to have a similar number of cells in the two groups, the celiac group and the control group.
Thank you, Luigina. Okay, so next question is from Mark Diaz. Would it be possible to fix the samples before single-cell RNA-seq pipeline in order to process together samples from different time points?
Probably I can answer that. It depends what type of fixation. So we've had experience with methanol fixed cells, and that differs sample to sample. So it has also to be tested out. So for some people it worked, for some people it didn't work. Otherwise, like formalin fixed, unfortunately, no, that wouldn't. It would damage the genetic material, the RNA, too much. What we offer is, of course, the buffer, which can preserve the samples for up to 72 hours. Otherwise, freezing, but then it would be single nucleus. Or doing the dissociation and cryopreserving, that also has to be tested on a case-by-case basis, just to make sure that the cellular representation is not affected by freezing thawing.
Great. Thank you very much, Khalil. Okay. Next question is from Nazia Pathan. Could you please elaborate on the quality control steps at the bioinformatics level?
Again, for this question, I will ask help to my colleague.
I can maybe answer in general. I don't know. In this case, of course, I didn't do the analysis. But usually what you are looking for are filtering steps to reduce outliers. So you remove any low expressing cells, like low genes per cells, because this might be a damaged cell or background. And then you remove also highly expressing cells. That could be doublets. So anything that is out of the range, out of the norm, where you might think that you got 2 cells in one well or multiplets. As well as the mitochondria, so you gate for mitochondrial content. Any cells that have a very high mitochondrial content are probably damaged and they have leaked their genetic material. Plus, you can add some steps more, for example, for removing red blood cells, so you do it on hemoglobin. If you're working with plants, there are also other types that you can do.
Great. Thank you, Khalil. And just as a reminder for everyone, if we can't answer your question right now, we will make every effort come back to you with an answer after the webinar. So next question is from Estefania Pavesi. How do you deal with impurities after tissue dissociation?
It depends on the protocol used. In our case, we dissociate the tissue by using enzymes and by pipetting, and by using also the temperature, because we put the dish with the samples at 37 degrees, and then when this step needs 45 minutes more or less, and then we filter the solution containing the dissociated tissue, and in this way all the impurities and all we are able to remove the impurities. I don't know if Khalil wants to add something.
Yeah, actually, I can add maybe something. So impurities have different causes, different origins. So what we do usually in the lab, we check under the microscope. We also do double stainings, AOPI, and if we notice that there are some subcellular, I would say, so we base that on the size and the fluorescence, we can remove them. There are some kits, for example, based on the cell of origin. If you are working with brain, you will notice a lot of myelin impurities that can be removed by bead-based kits, or you can also do differential centrifugation and remove those background. Plus, red blood cells can also be removed if you are working with a tissue that has a lot of blood, like heart. There are red blood cell lysis steps that you can take.
I can add one more thing, that it is possible to remove also dead cells by using a specific kit to remove dead cells, because after the dissociation, you can check the suspension to see if the cells are alive or dead, and if the dead cells are too much, you can remove by using a commercially available kit to do this.
Good point.
One of the things I love about this webinar is it looks like that work might be going on just behind Luigina right at this second. So no, thank you very much for that thorough answer. So our next question is from Ahmed Dawood. Did you use a transcriptomic atlas to annotate cells after clustering?
Okay, yes, we tried to annotate cells. We did hard work to do this, and we read a lot of papers about the intestinal tissue, and so we did it manually, and we also use atlas available online to annotate cells. So.
Great, thank you. And a question from Matthew Taylor. Is it typical to only have N = 1 sample per condition for a single cell RNA-seq experiment?
I mean, if I can say something, because also when you make this kind of project, you have also to considering the biological question that stay behind your project, and maybe if you look for bulk RNA, maybe in that case you have T0, T1, and T2 for the analysis. So maybe to give you a more statistical strength for your project, you need to have 3-4 samples. I mean, it's always depend about which is your final goal of the project. Maybe if you wanted to have a first screening about what is happening in the tissue that and this district tissues that you have selected and you are working on, maybe 1-2 samples could be enough, but take in mind that it will be just a preliminary vision. If you wanted to go more deeper, and especially if you wanted to give more statistical strength of your result, I mean, it's always up to do the researcher to do that. But of course, it will be need more samples to give more statistical importance of the result. And plus, keep in mind that after the library construction, there will be the sequencing. And also an important part that I think that we have to consider is the throughput per samples that you have to produce in order to perform all the bioinformatics analysis and the result that Luigina has shown here. So maybe you can get the result using a throughput of 45 gigabases per samples. In this case, to Luigina, we provide in 90 gigabases per samples. What I would like to say is every project has different stories, but I think that you have to keep in mind, if, as I said, if you wanted to look at a preliminary vision or if you wanted to go deeper in your analysis. I don't know if Luigina and Kate would like to add something more about this.
Yes, I can add something, because I can say that one, there is a problem related to the cost, that this is not a cheap kind of analysis. So this is one problem. Another problem is that in our case, we work with patients, so we cannot have the same patient in more points. So and we cannot have a lot of samples. Usually, to perform this analysis, we collect 2 samples from each patient in order to have a sufficient number of cells to perform the protocol and to generate the libraries. And then another thing that we are doing is that to select very well the patients. So we are enrolling patients with celiac disease that in a way are similar from an immunological point of view. So there are different kind of manifestation of this disease, because we have some patients with celiac disease with a very high concentration of the antibodies, anti-tissue transglutaminase antibodies, and other patients that have these antibodies but with a low titer. So what we are doing is to have patients with a very strong disease, and on the other end, we have the control that we are trying to have, and we are trying to have control as healthy as possible, even if it is not feasible, because if a child has to perform an endoscopy, he has to be something. Sometimes we know in advance that the condition, the reason why they are performing the endoscopy is a functional disease that we can consider an healthy subject.
Great, thank you, Andrea and Luigina. Matthew, I hope you appreciate that very thorough answer as well. The next question is from Sophia Hennigan. Did you collect new tissue samples for the second round of sequencing, or did you have stored samples? If stored, how did you store them?
We do not store these samples because after this experience, we know that we cannot store this sample, particularly to perform this study in patients with celiac disease. So what we are doing is to collect the sample during the endoscopy procedure, and when we have the samples, we immediately go on with the dissociation protocol to obtain the cellular suspension, and then with the cellular suspension, we prepare the single cell libraries by using the kit from Jackscope's kit. GEX-COP kit from Singleron, and we now know how to work with this kit, and we prepared libraries, single cell libraries from 4 subjects more, to patients suffering from CD and 2 controls, and we sent the libraries to Macrogen a few days ago, and we are waiting for the sequencing data. So, just to resume, we not store these samples. We process these immediately as soon as possible, and I think that it is the best way to do this kind of analysis, because we know that transcriptomics can be affected by a lot of aspects. So I think that in general, if possible, this is the best way to work. If possible, you have to try to understand how to do.
Understood. Thank you. Next question from Sofia Lavista Lanos. How long did it take for sequencing and bioinformatics analysis?
In this case, so considering the fact that we receive the ready-to-run library from Singleron, which is the headquarters in Europe is based in Cologne, and so basically it takes more or less around 2-3 weeks of sequencing, and the bioinformatics analysis. Okay, we have also our department, but also Singleron can provide the analysis. I mean, I can speak from our side. You have to add 2-3 weeks more. I think that more or less, considering only the sequencing plus, I mean, when I say the sequencing, I don't consider the library construction. I'm just to specify, I considering just the sequencing when we load the ready-to-run library on the NovaSeq 6000 or NovaSeq X Plus. Yes, as I said, it's between 4 or 6 weeks, considering the sequencing and the bioinformatics analysis. I don't know if Kyle and Luigina also you would like to add the turnaround time for bioinformatics analysis. How long did it take from your side?
For us, some kind of echo. Yeah, on our side, it usually takes a couple of days. Like when we get a library of 8 samples, it would take us a couple of days to run the primary analysis. Now, the secondary, what Luigina is showing, yeah, that would take more time. That would take maybe a couple of weeks to do, or even more. But the primary analysis can be run relatively quickly on our side. Yes,
I agree. In our case, the hard work, I think that we have already done the hard work, because in our case, what was difficult to do was the annotation, so to identify the cell clusters. Now we have these data and this information, so we just have to collect the data, and then the differential analysis is much more easy. Will be much more easy, we think.
Great, thank you, everyone. Next question from Monica Kladowska. How can one determine the optimal trade-off between the number of samples, number of cells, and sequencing depth to balance obtaining relevant data with managing costs?
The question here would be, what tissue are you working with? What's your question?
Yeah.
It's, as usual, biology is very complex. So it depends on what you're looking for, what tissue you're working in, so for example, PBMCs are not very transcriptionally active. Therefore, you can get along with shallower sequencing. But if you're working with, let's say, cancer, for example, which is more transcriptionally active, there you have to sequence deeper. So it's really complex. It will depend on the type of tissue, what type of cells you're looking for. If they are rare, you have to target more cells in order to find them. So yeah, it's very hard to give a general answer. It has to be on a case-by-case basis.
Yeah, and if I can add something, this is why when you would like to outsource this kind of project, it's good to have a consultation meeting from a Singleron side, from Macrogen side, because having a joint meeting, as we did with Luigina, it's important to put on the table every kind of issues and question to find which could be the best way in order to manage the tissue, which kind of library we can select, and plus the throughput that we have to generate for each kind of samples.
Yes, I agree. I think that it is crucial to discuss about the project, because everyone has a specific expertise, and if you add different expertise, you can find the way to understand how to do. And yes, that's it.
Got it. Thank you. Next question from Meng Zhu. How many samples would you include in each condition in your larger scale experiment?
Okay, we in our study, we will perform this kind of analysis in a total of 10 subjects, as I showed. And in particular, we want to have at least five patients with celiac disease and 5 controls to perform a differential analysis to find some differences in the intestinal tissue of patients with celiac disease and controls. And we, from each patient, we collect 2 intestinal biopsies. And in this way, we have seen that we can have the right number of cells to perform this protocol. We use the kit that is able to capture 10,000 cells. The maximum is 10,000 cells. And to capture this number of cells, you need cell suspensions containing 30,000 cells, if I correctly remember.
Great, thank you. There's lots of questions, which is great. Thank you so much for your interest, everyone. Next question is from Kate Bresner. Sorry if you've already covered this. What library prep was used?
I think that Kyle can reply for this, but the library was the Gexscope library provided by Singleron.
Exactly. So it's part of the Gexscope kit, and they are Illumina compatible libraries.
Thank you. Next question is from Sandrine Cabaret. In your original project design, you mentioned that your controls are patients with minor digestive disease. Why didn't you plan to take samples from healthy subjects as controls? And do you plan to control for a possible bias from the usual diet of the patients and controls?
We decided to include patients with minor intestinal disorders because, as I already said, our objective is to have to know how is the intestinal tissue from patients that are more likely and similar to an healthy subject, in order to see specific differences in the intestinal tissue of patients with celiac disease. This is, yes, the diet could be a bias, but we have to start in a way, and we cannot select patients on the basis of the diet. At least they are patients living in the same geographic area, so probably the diet could be similar. And then they are children, so usually children have to eat every kind of nutrients, so they cannot be vegan or something else.
Great, thank you, Luigina. Another question from Matthew Taylor. Is it important to be very specific on the exact location the tissue is sampled from in each patient?
Yes, we take the samples always in the same part of the duodenum, because in the duodenum you can take samples from the bulb, the proximal duodenum, or in the distal duodenum, and we take these samples always in the duodenal, in the distal duodenum, not in the proximal one.
Got it. Thank you. And I think this is the final question, and this was pulled out of the chat, so it's not in the questions tab, but a question from Ahmad Haidari. Was the stage of the disease considered in your study?
Yes, they were, regarding the patients with celiac disease, they are all patients at the diagnosis, so they are patients that are consuming and eating gluten. So they were suffering from an active disease.
Yeah, brilliant. All right. Well, thank you so much to our speakers for getting through all those questions so efficiently. I think we're like 1 minute over. So yeah, that's really all we have time for today. So thank you to you all for joining us. Really appreciate it. Don't forget, this webinar is available on demand, so you can watch it back at any time. Please do encourage your colleagues to register as well, if you think they would find it useful. And again, thanks to Luigina, Andrea, and Khalil for speaking and taking the time to prepare your presentation and your contributions. Thank you to our sponsors as well, Singleron and Macrogen, for making this webinar possible. And that's it. Hope to see you all again soon. Have a lovely rest of your day, everybody. Thank you.
Thank you.
Thank you.
Bye.
Thank you.
Bye.
Bye-bye.
Bye. Bye.
Bye.